Cell signaling and cell communication is really important in biology because this is the way how cells get information about their surroundings, respond to cellular environments, and communicate with each other. This is the cell, and in the membrane it's embedded a receptor molecule, usually protein, that has a very specific binding site, very specific for a ligand. The ligand is going to come a signal molecule released by other cells or by itself, the same cell, depending on, but it's very specific for that type of receptor molecule. It's going to bind, and this binding is going to change the configuration of the receptor molecule. So this step in cell communication, cell signaling, is called the reception. The receptors for the reception can locate in the cell membrane, and they are the transmembrane receptors. And we need the cell membrane receptors because some ligands cannot pass the membrane because of the hydrophobic, hydrophilic characteristics of the molecule as well as the cell membrane. And we will target 3 of the transmembrane receptors. The first one is going to be the G protein-linked receptor. So the receptor is linked to a G protein. The receptor is embedded in the membrane, and it's attached to a G protein. The G protein is going to get activated when the ligand will bind with the receptor. Here is the G protein. So at the binding, the GTP will turn into GDP. This is the energy for the G protein. And the G protein is going to move to an enzyme, and it's going to change the enzyme configuration, and the enzyme, activated enzyme, will start to provoke a cascade reaction that the cell will respond to that signal. The other one is the tyrosine kinase transmembrane receptor. It's a dimer. It contains 2 parts. The 2 parts are separated from each other when there's no signaling is going on, but when the ligands are attached to the tyrosine kinase, the 2 parts will join to will get phosphorylized through ATP. Phosphorylized, that means it will pick up a phosphate group from ATPs. They move towards towards each other, and they activate other molecules as they become activated through phosphorylation. So now the dimer will activate other molecules. And it will be the second step, the transduction, but we will discuss that later. So the third type is the transmembrane receptor, is the ion channel receptor. It's still embedded in the membrane. It contains 2 parts. Actually, so it's usually closed, but it's usually closed. See how it closed? And now if ligand is joined at a certain point, the specific binding site, the hook will open up and will let the ions flow in or out depending on. Now, G protein-linked receptor happens with certain neurotransmitters and hormones. The tyrosine kinase usually this is a receptor for growth factors, and the ion channel receptors are for neuron signals and neurotransmitters, basically. Neurotransmitters will open the ion channels. So that was in the cell membrane, transmembrane receptor. The other type of receptor is a receptor inside of the cytoplasm, so cytoplasmic receptors. These receptors are in the cytoplasm because the ligand can cross the membrane, so it is hydrophobic, and it can go into the cell, inside of the cell. These are the steroids and the thyroid hormone. Now, what is happening, what is going to happen at this point? Here is the cell and the nucleus. This is the nucleus, and we have a intracellular receptor molecule, and the steroid, it's going to pass the membrane, goes and binds with the intracellular receptor molecule, forming a hormone receptor complex. And that complex will go, this is the complex, will go into the nucleus and acts like a transcription factor, activates a certain gene in the DNA. So we covered the reception. Now let's talk about the next step that happens inside of the cell. These three blobs represent different molecules, proteins or other molecules, and the arrows represent many steps, and these many steps are going to add up towards the transduction, the second step of the cell signaling or cell communication. The transduction is a series of steps after each other, one kind of a domino effect if we are thinking about in a linear way, but it can also amplify, so it's a series of steps, but it also can amplify signals. It is great for coordination and regulations of the responses. Also, it can involve second messengers. The first messenger is usually the ligand. We call it the ligand, or the ligand. The second messenger is, so the secondary messengers can be the cyclic AMP and the calcium ions. And it can involve also protein kinases. So the secondary messengers are really important to amplify the signals. One signal can have a huge outcome response from a target cell. So the third part is the response for the signaling and the communication. The signaling the response can be like activating an enzyme, or the ligand message is to rearrange the cytoskeleton's structure, or activating specific genes and producing protein synthesis, different protein synthesis, or changing in membrane permeability.